Hello and welcome to ChatGCP, a medical science, clinical research and commercialization podcast with me, your host, Charles Beasley. On today's episode, we have Dr. Malvindar Singh-Bains, a research fellow at the Centre for Brain Research at University of Auckland. Welcome.
Thank you.
Welcome to the chat.
It's a pleasure.
Great to have you on today. So you work in primarily neurodegenerative diseases,
Yes.
such as Parkinson's, Huntington's. What got you interested in this research?
It all started when I was a young fledgling. No, just kidding. When I was a kid, I had quite an interest in getting my hands dirty. So I was always outside trying to make little curries in our backyard by throwing a whole lot of water into giant holes and chucking leaves into it. So I always had this inherent interest in gross stuff from being a kid. And then when I navigated high school, I guess I was interested in a lot of different subjects, but human biology was definitely one of them. And it wasn't until I approached university when I experienced a set of lectures by my now mentor and boss, distinguished Professor Sir Richard Fall, who spoke about the human brain and its complexity and how it's the last frontier. And there are so many neurological diseases that we yet don't have cures for, and that really stimulated my interest. But I think from a more personal perspective, paralleling my scholarly life and going through university, at the same time, my paternal grandma was diagnosed with dementia after having a series of strokes. And she used to live, because she's now since passed away, but she used to live in the United States. And so every couple of years I would visit her, and I saw her decline over time. And I think for me, as a granddaughter, it was difficult to walk that journey with her, and it was about a 10-year journey. And in addition to that, my maternal grandmother, I didn't have the opportunity to meet. And she passed away with dementia when I was 2 years old. So I already kind of had a family connection with neurological disease, and I guess that also stimulated my interest in wanting to know more about it. And perhaps if I could contribute to some sort of breakthrough or some sort of therapeutic intervention later in my life, that might in turn impact people that are suffering today. So that was kind of what got me into neurodegenerative diseases. But I always had this fascination with gross human biology, blood and guts in the brain. Yeah.
Love it. Is it the final frontier aspect of the brain that is particularly interesting to you? Or that there's, so much, like you say, so much unknown. Was that appealing as opposed to some of the other parts of the body which are a little bit more well resolved, shall we say?
Well, if you have a dodgy liver, you can essentially replace part of it. Liver transplants are becoming more and more common. Likewise with lungs, likewise with the heart. If we think of ourselves as a car, if our parts start to go, we can replace them over time. Whereas with the brain, to date, if a single part of the brain is ailed, if there's a part that undergoes some form of disease, you can't reverse the progression of that disease. You can't really fix it once it's sick. And this is probably the only organ in the body where you can't really fix it. In one instance, if you've had a stroke, for example, and it's early enough, you can bust that clot and hopefully you've prevented brain cell death. But in most other instances, particularly with neurodegenerative slow progressing conditions like Parkinson's or Alzheimer's or Huntington's, once that starts, you can't reverse it. And I think for me, that is what the final frontier is that if we're able to harness the prospects of trying to regenerate a sick brain or trying to reverse sickness in a brain, then we've passed that last frontier. So with every other organ in the body, as far as I am concerned, you can do something to repair it. With a brain, you can't yet. So this is why I'm in the game.
Nice.
Yeah.
Oh, it's absolutely amazing how little we probably, on the grand scheme of things, how little we do know about the brain, its complexity. We could probably dive into the realm of consciousness, but I'm never going to go down there with this kind of thing because we could just tie ourselves into knots.
Well, I always ask myself, is the mind in the brain or the brain in the mind?
Oof,
right?
Deep.
Yeah, very.
Yeah. Am I my brain or is my brain me?
Exactly.
Yeah.
And then where is your soul? Yeah, so these kinds of questions, I think, have plagued, society for generations, right? And we still don't have a proper answer. And so I think as a biologist, I find all of this quite fascinating. And we're talking about consciousness, for example. What is consciousness? Where does it come from? Where's it born?
Yeah.
What part of the brain's
I mean,
responsible
the
for that?
reality we see does not exist. I mean, this table is not actually a table. It's just a series of protons and electrons. Yeah,
Arranged.
giving off a wavelength that we perceive.
Yes.
I mean, you could literally go mad thinking about that, I feel.
Yeah.
But you raise a really good point, and it's no more that we hear often about prevention is better than the cure.
Yeah.
And in no part of the body is that more obvious or more relevant than the brain, right? So maybe to frame this, how do we keep our brains healthy? How do we prevent brain disease, and what kind of categories can we put brain disease into, shall we say?
Okay. That's a big question, but I guess if we look at a lot of these degenerative conditions like Alzheimer's, for example, there's a window of opportunity we have where you could develop an intermediate situation that looks like mild cognitive impairment, which is what we consider to be not dementia yet, but the area where things might start to go, like where you start showing elements of forgetfulness, right? If we can prolong that period so it doesn't become dementia, in other words, if we can hold off the onset of dementia by at least 5 years, then it's likely the patient can live long enough to have a relatively good quality of life and probably succumb to some other condition which is quick and painless, right? So if we're talking about trying to maintain a healthy brain, especially in that period perhaps of mild cognitive impairment, there are some things you can do. I think if you treat the brain very much like how you treat your heart, those risk factors for heart disease become just as important and as apparent if you're in this mild cognitive impairment category. And so if you think about brain training exercises, for example, like challenging your mind with crosswords and Sudoku puzzles and the like, that's going to stimulate neuronal firing, so the firing of your brain cells, right? And that will encourage you to think critically and put some energy into your brain activity. And that strengthens the connections between your neurons and reduces the likelihood of them dying. That's one way. So, those type of brain training exercises.
Use it or lose it type things, right?
Absolutely.
Yeah.
And you don't need to spend, a lot of people say, I don't want to spend tons of money on getting some weird computer game or brain training. A lot of people start Googling brain training and then they start finding these very expensive games and things. No, it's as simple as just taking that Sudoku puzzle and giving it a go. Use it or lose it. I think the other thing that's really important is environmental enrichment. So some studies suggest that if you have a group of mice and you take one poor sod out of that group of mice and leave them on their own, they become very sad. They stop interacting. They may stop eating, and actually their cognitive impairment worsens. Whereas the group that are all sort of together, they're being more social, they're interacting, their environment is being enriched, and they are less likely to develop memory-related symptoms. So if we take the mice, we now apply that to humans. I think if you're encouraging social interaction with people, and this is why, for example, rest homes and respite care facilities are actually quite good on that social front. If you encourage interaction between people, that stimulates neuronal firing as well. And at the same time, if you experience novelty, so for example, if you go for a walk in a place that perhaps you have never been to and you take in all those different sights and sounds, that's going to encourage what we call plasticity, learning and memory to take place. And so I think environmental enrichment combined with social interaction and taking into account what's good for the heart being good for the brain, those are some of the basic things we can do to stave off dementia and to improve our cognitive abilities without having to break the bank. And one more factor is very important is exercise and actually utilizing your body's muscles. What that does is it boosts oxygen to the brain, it boosts your metabolic activity, and it encourages the release of those happy chemicals, your dopamine, your serotonin, and therefore it helps to stimulate positive thoughts. And I think that has an impact on cognition as well and the ability to recall memories. And so I think, some of those factors are really important for overall brain health, not just, when we're perhaps older and at risk of dementia, but even in our, young lives, 20s and 30s, when we are, getting used to those 9 to 5 kind of jobs and we're starting to develop a sedentary life. We're not necessarily eating as well as we should. Working from home, so we're actually doing all those things that reduce brain plasticity, starting to apply those factors now while we're young. And then hopefully what will happen as a consequence, as our population ages, hopefully dementia will develop later on instead. And, we will have something else that will take our lives that's just fast and non-progressive.
Well, you raise an interesting point, because how much of the brain is dedicated to controlling movement, so skeletal muscle? I mean, so that's the interplay with the brain and exercise, right? I mean, if you're not using it, you lose it.
Yeah.
And if you're not using it to move your bones and move your muscles, move your bones, you don't know what I mean, guys, then that's a huge part of your brain, right?
Yeah, it is. I think everybody likens the brain to it being segregated according to continents, which have a certain function. But our brain operates as circuits. If you can think about it in terms of we have parts of the brain that are interconnected with one another that carry out the fundamentals of movement. If we think about movement on its own, we have the motor cortex, which is in the frontal lobe of the brain. That's our main area responsible for movement, but that interacts with the cerebellum, which is at the back of the brain. It has the highest concentration of neurons, but that in turn interacts with a bunch of structures in the brain responsible for the initiation and carrying out of movement called the basal ganglia. So you have an entire loop that connects the motor cortex, the basal ganglia, the cerebellum, the thalamus, that form this interconnected circuit. And when part of that circuit is destroyed, then you develop disease. If all of that circuit is destroyed, then you're going to have real difficulties in moving. And so that's just movement as a whole, but we have similar circuits that play out for the development of pleasure, the development of pain, the development of memories. And so everyone talks about the hippocampus, the part that looks like a seahorse in the temporal cortex of the brain, that's associated with housing your memories. But what we now know is that that's in fact part of a circuit responsible for the generation of memories. And so if we think about the brain as like a bunch of highways, that's how we need to sort of understand how it works. And we still haven't cracked that puzzle entirely. Researchers are discovering new components to these circuits that are responsible for, say, movement or cognition.
Yeah, because it's very easy when you're learning these things to go, we're just going to pass out the parts of the brain into different structures, different parts of the topography, parts of the map, parts of the globe, whatever. Yeah. But yeah, I mean, it's all integrated. I mean, it's not as though our body is just discrete packages of these kinds of structures. They're all intermingled. Yeah. Yeah, and no more true is that than of the brain, because it is so complex.
Absolutely. Yeah.
So we're social creatures. We need that social stimulation. We need novelty. Yeah. We also need a good diet and exercise. Is that a bit why we often hear talks of people really having like quite stark declines after retirement or after the death of a spouse, for example, because the social aspect kind of goes away?
Yeah. So you're 100% right. So I think the element of self-neglect that happens in response to being in an isolated situation or experiencing extreme grief, it certainly has an impact on all those different parts of well-being that we have. So obviously, if you've become isolated, if you're no longer going out, if you're no longer interacting with people, if you're no longer eating right, that's going to have an overall impact on how you think and how you respond and how you process information. And so this is unfortunately why a diagnosis of dementia in itself can lead to dramatic decline, because the element of grief associated with that, then the patient starts thinking, if this is what's happening to me, why should I bother making any changes? But I think the evidence now suggests that making these changes means that progression from impairment through to dementia can be longer. And this is why when I'm speaking to people that are in the dementia communities, it's about living well with dementia. It's about doing all the right things to ensure that progression, if it is to progress through to dementia from mild cognitive impairment, is as pleasant as can be. And that means you've got to have your people around you. You've got to have a support network in place. You've got to have a community. It means you have to have a meal regime. It means you need to schedule an exercise. It means you need to have some form of environmental enrichment, because I think what happens as a consequence is that these patients can have a pretty decent quality of life after diagnosis. And this is what we're trying to push from a more lifestyle perspective, particularly in my community, like the Southeast Asian community, if we touch on that. The term dementia has so much stigma associated with it that often a diagnosis leads to further isolation. You end up feeling a little bit ostracized from your community, because now suddenly people don't know how to interact with you anymore. Are they remembering what I'm saying to them, and all of that? So I think if we can just change that narrative and talk about the wellness side of things, we might be able to actually have a direct impact on the outcomes for some of these patients as well.
And with the social side of things, does this need to be in person? I mean, we're in a very, and increasingly more so, digital age, and there are digital health initiatives, a dime a dozen these days. But how important is the actual physical, social aspect?
I think COVID has really dampened and made it difficult for us to have more in-person engagement. And now that we're coming out of that period, as a researcher, I think people are less inclined to approach a university institution for advice from researchers or clinicians or people in their fields. Whereas what we need to do as advocates for positive brain health is to get out of our ivory towers and actually get out into the community again. And so a lot of my passion is actually going out into rest homes, or in the case of the Southeast Asian communities, getting out into temples, mosques, gurdwaras, and actually speaking to people within these communities about the importance of brain health and how we can adapt lifestyle changes in a more culturally appropriate way too. Because I think people forget that there are certain lifestyle factors that you may not be able to change because culturally it's just the norm, right? And so I think by getting out in there and having a korero with the community, we're going to have more change. So I think the digital age, yes, while it has its benefits perhaps for engaging with young people, I think for the generation that is closer perhaps to retirement age, that type of communication doesn't cut the mustard. In fact, it's probably worse, and it actually makes it more difficult to engage with those communities. And so I have definitely witnessed the power of face-to-face communication, particularly when you're outside of the cities. So if you're going out into the regions, like I'm just thinking up north, they really appreciate researchers, scientists, clinicians, experts in the field going out to see them, and they're not gonna chime onto a Zoom call with you, like. For them, that's not engaging. They'll skip the call. But if you go out there and you have a meal with them and you have a general conversation with them and also allow questions to be asked, I actually think there's so much power in that level of engagement, and we underestimate the power of that as well. Definitely. It's the same impact of in-person teaching in schools.
Oh, gosh, yeah.
Right? Yeah. If your teacher is in person and interacting with you, you're going to engage so much more than with a screen. People are desensitized to screens. People have 4 screens going on at the same time and are still not satisfied.
I mean, I've got one going right now.
Yeah, we've got how many screens? Still not satisfied.
Yeah, absolutely.
Yeah.
I mean, it's bizarre. The older I get, so old, the more I realize that, man, we can't re-engineer ourselves for this age that we've created for us. We've got digital everything, super convenient. I have access to so much information. I can talk to people on the other side of the world without even breaking a sweat. Yeah. But is that good for me if I forego what is naturally appropriate or biologically appropriate or what the imperative that my body has?
Yeah.
So like the in-person social aspect, lifting things, walking around, not running because I hate running.
Who likes running?
Yeah, weirdos. But yeah, I mean, we can't innovate digitally our way out of fundamental biological problems.
No. We are creating more problems for our biology through increased digital communication as well. Just the number of students and staff I have around me that need to go to physio because XYZ is, you've been sitting for too long or you've got a knee issue and what have you. It's all catching up with us now. And I'm just, these are people that are in my age group and, I'm not a day over 19. So obviously, I think if we look at then the senior citizens, it's going to be more difficult, right? So yes, there's an aspect of, you don't want to fall behind and not be part of the tech age and not be part of that level of engagement and communication, what have you. And also digital Sudoku puzzles, right? Digital Sudoku puzzles, pretty cool. And that's all fine. But I think the power of face-to-face, in-person communication, of narrative in that regard, that is what is going to change people. Why do you think politicians go out into the community during election time?
Exactly.
Yeah.
Exactly. And only once in every 3 years, right?
Yeah.
Yeah, it's interesting. I just feel like as I said earlier, we can't digitally innovate our way out of fundamentally biological problems. No. And COVID for the first 6 months was probably hunky-dory. Everyone felt good. Those that could work from home did. And it was like, look how productive I am, but what are the background consequences?
Yes.
I mean, I've been working from home for 3 years, which has been fantastic. I've got a 3-year-old daughter. She turned 3 a week ago, and she has never known her dad not to be at home, which is awesome for her. But, after 3 years, you struggle to be able to make eye contact and string a sentence together without sounding like you've dropped a few IQ points. As you can tell. So it's really interesting. So why don't we pivot to your area of research? So neurodegenerative diseases, so specifically Huntington's. I've heard you call Huntington's is a model disease that needs to be studied and can influence or inform potential avenues of research and treatment for other neurodegenerative diseases.
Okay, so let's talk about Huntington's. It's a progressive neurodegenerative condition, which means there is a loss of brain cells in certain areas of the brain over a period of time. This disease manifests typically in midlife, so sort of age 35-ish, around that point in time. And because it's a genetic condition, it means that if you have the gene that causes the disease, you have a 50% chance of passing it to your child. That doesn't mean that if you have 2 children, one will get it and one will not. It's an independent 50-50 chance. So what's interesting about this condition, and the reason why I'm still, it's been 13 years since I've been started studying this disease, is that it has a myriad of symptoms. And you have symptoms that range from the cardinal symptom, which is chorea. So chorea is a movement disorder, and chorea is Latin for dance-like. So you have some patients that develop this kind of this dance-like writhing, convoluted movement.
Like lilting.
Lilting, yes, and there's an inability to stay still. To be honest, that symptom doesn't bother the patient. What bothers the patient is the cognitive and psychological symptoms. And this is why this disease is so interesting. So you've got, key psychiatric symptoms that are often associated with other disorders like schizophrenia, where you have hallucinations and what have you. You have a mood disorder that, results in potential aggression, inward and outward irritability. So you have, quite a few interesting psychotic symptoms in there. But on top of that, some patients can develop a cognitive disorder, so loss of memory, loss of ability to retrieve memories, loss of recall, loss of being able to remember new things, confusion, and what have you. So what you have is a disease that has a little bit of all the other diseases that we know about.
Right.
And because this disease is progressive and is associated with a loss of brain cells, that also shares homology or similarities to other conditions, including Alzheimer's and Parkinson's as well. What's interesting on top of that is in the brains of these patients, over time, there is the accumulation of these sticky proteins. And in the case of Huntington's disease, as the name suggests, the name of the protein that gets sticky and accumulates is called Huntingtin. So it has a loss of brain cells, a myriad of symptoms, buildup of disease-causing proteins. This is very similar, if you think about it, to what Parkinson's is. In the case of Parkinson's disease, you have a buildup of alpha-synuclein, which is the type of protein that gets sticky and builds up in those brains. In the case of Alzheimer's disease, it's the two proteins beta amyloid and tau that accumulate in the brains and form these sticky deposits. So if you look at all of these conditions, Alzheimer's and Parkinson's, for example, are quite, and motor neuron disease, and a number of different neurological conditions, are quite needle and haystack disorders, unless they have a clear gene that causes it. In the case of motor neuron disease, you can have some types of genes that lead to the symptoms. With Huntington's, it's the same gene. It's the same gene it's essentially called Huntingtin as well, that causes the disease. So what we have is a clear therapeutic target, a target to treat all of these symptoms, as well as the brain cell loss, as well as the deposition of this protein. So for me, studying Huntington's is almost like studying a little bit of all of these conditions at the same time, which is why I've stayed in this realm. A lot of people ask me, well, it's a rare disease. Why are you studying a rare condition? Well, it's not as rare as you think it is. And if I reflect on all of these different symptoms, you realize that it has overlaps with other diseases. So actually, we're informing these other conditions through studying Huntington's. So this is why I've stayed in this field and why I'm quite confident that at least in my lifetime, we're going to have a genuine breakthrough because we have a clear target.
Yes.
The challenge to the target is that this target is responsible for our basic functioning of life. So you and I have the gene for Huntingtin, but we have the gene that is responsible for our daily function. It doesn't cause the Huntingtin protein to misfold or do weird things, whereas in a group of the Huntington's patients, that's what it does. And so I think that if we have a medical breakthrough in Huntington's, we're directly going to have something that could extrapolate to the other conditions. And this is why I'm so excited to be in this field. And actually, I think this is why we're getting quite a lot of interest from drug companies and therapeutics in the Huntington's disease field, because they can see that it's going to have a direct impact on some of these other conditions too.
Yeah, amazing. And in terms of a patient's potential progression, it's not like a one-size-fits-all type of thing, is it? There's a real spectrum in terms of age of onset, severity, or how quickly it progresses? Yes.
So even if two patients are siblings and they have the same genetic mutation, so just to give context around this mutation, I use the word mutation just willy-nilly there, but what happens is there's a gene defect, which in the Huntingtin gene, which results in a faulty signal that comes from the gene. And that faulty signal is called a CAG repeat. So we have a code which forms from our genes to subsequently transcribe and make proteins, and these proteins do things in the body. And so with Huntington's disease, we typically have the CAG repeat length that is less than 35, typically 35 repeats when we don't have Huntington's disease. And that means we get normally producing Huntingtin protein, and it carries out all of its normal functions that's involved in cell survival, that is involved in normal cell division, it's involved in the clearing of normal proteins. It's essentially every single process in the body Huntingtin is involved in.
So it's not like you can just get rid of Huntingtin, the protein, and
nope.
You're sorted.
If you knock it out in animals, the animals die 3 days from birth.
Wow.
That's already been trialed. We've trialed it lots of things. So because animals contain the Huntingtin gene as well. Or even if you've add in the human Huntingtin gene into some animal models. Some studies have tried to just completely knock down that gene. The animals just don't survive. And so we know it's important in cell survival. Whereas in patients that have Huntington's disease, that sequence repeat from the gene is typically longer than 35, or definitely longer than 40 repeats you'll show the disease, but typically longer than 35 means you have a chance of passing it to the next generation as well. And so that's how the disease is caused. What my research has sort of uncovered is that this single gene mutation, even if it codes for the same length of CAG repeats, even if the patients are siblings with the same gene mutation, they have often different symptoms. And we believe that and this is also backed up by my own research, we believe that the symptom generation is actually the product of the pattern of brain cell loss across the brain. So what we've done is we've mapped certain cell loss patterns in the brain and found that depending on the type of symptoms the patient has, then subsequently depends on where the brain cells have died. So we can determine that post-mortem. So we study the brain tissue samples from patients who have died with the disease, and we've discovered that certain parts of the brain die off that are indirectly responsible for the symptoms. What I don't have the answer to is how does the action of the gene result in the pattern of cell death? And that's now what my research is trying to work out. So I'm trying to map out the deposition or the buildup of that disease-causing protein to see if we can find a link with the potential cell loss patterns. Because if we can find that link, then I think we have sort of an idea of how to develop therapeutics into the brain. Because you don't want to develop a therapeutic and deliver it to the brain to places that don't need it. Because as I mentioned, Huntingtin does normal things. And if you're trying to clear Huntingtin, you don't want to clear it in parts of the brain or body systemic circulation as well, where we need Huntingtin to do its thing. So my research is twofold, trying to work out the pattern of brain cell death and how it links to the deposition of that bad protein, to then account for how the symptoms are generated, and two, to work out what parts of the protein we need to live versus what parts cause the buildup of the protein. What gives it that greater propensity to stick together and build up in the brain? So that's kind of where my research is going now.
Gosh, so much to unpack there. Well, I love it. So if I can summarize, and I'm going to butcher this, but bear with me. So the CAG repeats create misfolding of the protein. Do all of the proteins get misfolded, or is it just a likelihood based on the number of those CAG repeats that you have?
A likelihood based on the length of the CAG repeat length, and also not all of them misfold in the same way.
Okay.
And not all of them form the same types of chunky aggregate bits in the brain either. There's different kinds.
So it's like there's a lot of gene therapies that are coming on the market now for various different diseases, like small interfering RNAs, and they knock down the expression of specific mRNAs, so you don't get the aberrant protein. But you can't really do that in this context because you need some of it to maintain cell survival.
Yes. So if we talk about the main gene therapy that has been trialled in patients, the tominersen trial or antisense oligonucleotides, that first trial did not discriminate between normal and diseased Huntingtin. So that trial was targeting all Huntingtin.
So it's like throw it against the wall and see what sticks kind of thing.
And so while in theory what they were suggesting is that, you know, if we just turn down the gene enough by a little bit, would then slow down the production of the bad protein, and hopefully over a period of time we would see a slowdown of symptoms. What was not done was discriminating bad Huntingtin from normal Huntingtin. And so subsequent studies are now trying to work out how to target more closely the pathogenic or disease-causing Huntingtin protein. But that didn't happen in that trial.
Is that because, you know, there's always a time lag between fundamental science done by people like yourself and what is done in industry, they might be a few years behind. Let's be generous. A bit longer than that.
No.
Is this like an old school approach to
Absolutely not.
Huntington's?
I actually think the trial, in my personal opinion, I think the trial wasn't long enough. But typically a clinical trial is only 2 years long.
Mm-hmm.
The science to get to this point was quite robust, but it just has one serious issue, which is we haven't isolated the bad guy only yet.
Yep.
That's it.
Yeah.
And so instead of trying to work out exactly what the bad guy is, let's just focus on the general target and hope that by knocking down it generically, it would have less of a propensity to stick to one another, and hopefully enough of it would just get dialed down so that the bad guy doesn't form as much, but we have enough good guy doing its job.
Right.
That's kind of the medium they were trying to achieve. So the science was conducted in an animal model first, then moved to a larger animal model, then had a small cohort of living human participants, and that trial was more focused on safety. And the trial, the drug was delivered via a lumbar puncture. And so
Because it's hard to get into the brain, right?
try giving a lumbar puncture to a Huntington's patient with chorea. That's quite challenging, right?
Yeah.
So some of the safety issues around delivering a lumbar puncture to HD patients in itself is a big thing.
Yeah.
If you think about a clinical trial, patients continuously moving, right? There's a lot of challenges associated with this trial. I think the tech is really cool. I think it just needs to be packaged to be more specific for what the bad guy is.
Yeah.
And that's the fundamental science that I'm working on, is what is the bad guy?
Yeah. And you kind of alluded that there's not just one bad guy. They can fold in so many different ways.
Multiple forms of the bad guy.
How many bad guys are we talking?
So many bad guys. So one of my studies that got published last year was initially just finding the right tags for the bad guy. And so in human brain tissue studies, we often use antibodies to bind to proteins to tease them out from everything else, right? So I was just trying to work out what are the good antibodies, what are the good detection probes we can use to isolate bad Huntington. That was never really done.
Wow.
It's a fundamental, simple study. And so we profiled about 30 Huntington's brains in our brain bank to see whether we could target the sticky bad, what we think is... Okay, first of all, can I just add, the sticky form of the protein might not be the bad guy. What about the teeny tiny guys that just circulate around the blood, surfing through and just doing their thing? Like maybe they're the bad guys and we need to be targeting those. We don't even know what the bad guy is. So the sticky stuff is what's left behind. But what if the sticky stuff is a protective mechanism to stop brain cells from dying?
Yeah.
Right?
So that's a fundamental assumption that hasn't actually been tested yet.
Correct. So now the researchers in the Parkinson's and Alzheimer's and Huntington's community are thinking, oh no, maybe the bad guy is not the folded uppy stuff. Maybe the folded uppy stuff is a protective mechanism.
That would turn the whole thing on its head, right?
And it does.
Wow.
So you have a group of scientists now that are suggesting, oh, maybe the folded stuff's not the bad stuff. Maybe the folded stuff is protective.
Gosh.
Is protein aggregation protective? Are our bodies trying to sequester or like stick that together and then throw it in the rubbish?
Yeah.
Right?
Yeah.
Is that what our bodies are trying to do?
That I've always wondered about these kind of things. I mean, there's consistency in terms of the high level framework. You've got accumulation of tau or beta amyloid, or in this case Huntington's.
Yep.
That, because they're consistent across different diseases, you would kind of think maybe there's an evolutionary justification or reason for it.
Right.
Why would it be there if it was exclusively bad? And if it was exclusively bad, how has it persisted for millennia?
Right.
So
this is what I find fascinating as a neuroscientist, is that we're still trying to work out the molecular mechanisms behind these diseases, and this is why you can just sense that I'm so passionate about this being the final frontier, because I think if we can work this out, we've actually worked out most of the diseases.
Yep.
So you've got researchers that are trying to bust these big, sticky proteins, but then you've got researchers thinking, oh, maybe we need to slow down the movement of the small guys so that they don't traverse from brain cell to brain cell to brain cell, so that they don't form sticky things to begin with. So then when do we give the treatment?
picturing this, you've got a whiteboard behind you or a detective's corkboard, and you're going, who are the bad guys? And you've got maybe 1,000 pictures of blank faces, and you're trying to figure out. And maybe you'd flip, if you were like an all-powerful being, you'd flip them over and go, nah, he's not the bad guy. Why are we looking for him then? Amazing. It sounds so much infinitely more complex than I had really could have appreciated it.
And this is why I think, the community in the clinical world and the community in the biomedical world have to have a lot of dialogue, because I think we can help inform each other. And actually what's interesting is the community, so the patients impacted by the disease and those that are at risk, they actually have the biggest impact on our research. So I remember some patients saying to me, actually, you know what, the career doesn't bother me at all. It's not the movement stuff that bothers me. Do you know what bothers me? My gut. I have an upset stomach. And then did you guys see the giant brain-gut axis publications that emerged for links between gut health and Parkinson's disease, or links between gut health and dementia, and now links between gut health and Huntington's? The key to these diseases come from the patients. We just have to listen to them.
Yeah. And everything's linked. Nothing.
Well, we know the Huntingtin protein is present in all our organs.
Yeah, amazing.
So now they're looking at treating Huntington's in the liver.
Really? And if go back to something that you said previously, there might be different parts of the brain where the accumulation is correlated with symptoms or cell death. Yeah. Can you target different parts of the brain? You can target different tissues. Like I know for siRNAs, they use a GalNAc adjuvant that targets it to the liver, targets an siRNA to the liver. Can you target to the brain? I mean, you have to go through the lumbar puncture to begin with to get past the blood-brain barrier. It's hard to say, isn't it?
It's annoying.
But then can you target different parts of the brain?
You can. So you can join a lot of these therapies with viral packages that traverse only certain cells, and they only activate in certain brain cell types. So you can do a cell-specific siRNA. But in terms of developing therapeutics to certain brain regions, it would follow the similar process to how you would deliver deep brain stimulation. And so deep brain stimulation electrodes are delivered directly into the brain using a very small drill, and you can then target the therapy to that area. You need to then work out what the spread of the therapeutic is to that target area, and you can control for that by giving the right amount of drug. So it is doable. Has this tech been trialled well in humans? No. That's the next step.
Yeah, and also I'm pretty sure your research would inform a lot of whether that would be worthwhile.
Yeah. So
is the bad guy there? Do we know who the bad guy is? How much bad guy? Does it have downstream implications to the symptoms or the progression?
As a neuroanatomist, my model approach is to try and study all the brain regions as much as I can. So I started off studying just the central part of the brain, the basal ganglia, and then I traversed out to the cerebellum, and we discovered that Huntington's also significantly affects the cerebellum, which was sort of like a world first finding. So what that means is that we need to consider structures outside of the central circuit to treat. So actually, a lumbar puncture might not be a bad option to treat if enough drug can get through the blood-brain barrier into the brain through the cerebral spinal fluid. Because the cerebral spinal fluid, it bathes the brain, right? So if it bathes the brain, we can hijack that system. It's like hijacking a river, right? The problem is they're not getting enough drug in. So we actually need good cargo to deliver enough drug at the right concentration to the right parts of the brain. So I guess that's where my research informs how far to go. We definitely need to go out to the whole cerebral cortex for Huntington's disease. It's not a single region affected disease. We've rewritten the textbooks already. Yeah.
Because Parkinson's, that's one where it's like we know the region that we need to primarily go after.
Even in Parkinson's, there's a pattern. There's a pattern. While, the loss of dopaminergic containing neurons in the substantia nigra is the main target for Parkinson's disease. And that's realistically, if you're targeting that part of the brain and the patient already has symptoms, it's too late. There are parts of the brain around it that are affected too, just to a lesser extent. And we also know that there is a lot of research suggesting that Parkinson's starts through the nose, right? Through the olfaction system. And therefore, all those structures going through from the nose, through the cortex, leading to the substantia nigra are affected in the disease as well. And then you have all the researchers suggesting that Parkinson's starts in the gut. So then you have alpha-synuclein coming through the spinal cord, going up the brainstem, then heading towards the substantia nigra.
Right. So it's coming from another part of the body and accumulating.
Well, that's what a lot of the new research is suggesting, that you can find this protein through the pathways leading up to the brain from outside of the body. And so, once again, ideally, if we developed a therapeutic and just put it in the substantia nigra, you would want to deliver it before you lose dopaminergic neurons. So getting other biomarkers of a disease where things change earlier on, for example, maybe loss of smell, that would be quite a cool way to know when to deliver a therapeutic. Or if you do have changes in bowel health, because a lot of patients talk about their changes in bowel health before they have motor symptoms.
So going back to Huntington's, if you were a betting woman, what do you think would be the next big breakthrough in the case that would crack this open and open up some more targeted alternatives?
I think if we had a very targeted gene therapy that only focuses on the disease-causing part of Huntington, that will be the breakthrough. But to get to that point, I think we need to work out what the bad protein does. So I think we need a little bit more time on that to be really sure that the next time we put therapeutics into patients, that it's going to be turning off the bad protein and leaving the function of normal Huntingtin alone. So I think we're not far from that. Considering that the gene was discovered just over 20 years ago, when we had kind of nothing happen to the development of gene therapies in the past 5 years, I think we have a huge window of opportunity now. So I think that's where the big breakthrough is going to be. And I feel it. I feel it. And I feel it because we're getting more and more closer to the bad parts of the protein. And I just think we need better tools.
And what are those tools? So you mentioned you've got the antibodies.
Yes, we can use molecular probes of different kinds. If you're targeting it at the RNA level, then you're using what we call RNA-based probes. So it's using a technique called in situ hybridization, which detects RNA in tissue. You do have genetic targets. Like you can use sequencing to look at, but we know what the sequence of the protein is, right? But for protein-based discoveries, we use normally an antibody-based approach, and that's because antibodies are readily available. There are different kinds, and what we can do is exploit the different kinds of antibodies to bind to different things we're interested in tissue. For example, if we're interested in Huntingtin and its particular form in certain brain cell types, we can use multi-label antibodies on top of one another in the same tissue sample to be able to profile what the cellular distribution is of Huntingtin.
Right.
So that gives us a lot more information. And now we have the advent of something called multiplex immunohistochemistry, which means we can now profile up to 100 different antibodies in a single piece of human brain tissue. So you can bind to 100 different things. Wow. Imagine if we had 100 different targets for Huntingtin that we could probe at the same time. The only way we can do that is have good probes developed. So this is where I then plug on air to every company in the world to develop different probes to this protein, because what we'll then have is a great bank. So I actually work with a biochemist in Switzerland. He's one of my collaborators, and he is trying to develop better antibodies and molecular probes to this protein. And you have to realize is with these probes, they have to be able to penetrate brain tissue and get to their target. And Huntingtin is a sticky, annoying protein, right, to get into. And so there are a lot of challenges to developing targeted probes. And if we could throw money at that, I think we're in turn throwing money at therapeutics.
Absolutely. Amazing. And so these probes would go after distinct shapes of the Huntingtin and distinct clumps?
Shapes, different parts, different sites that are modified. So proteins have different things that they attach to, or they have different forms. So they might have a modification done to them. So, for example, we call them methylation, so the addition, for example, of a methyl group, or they might have an extra disulfide bond in the protein. So there's different things, or they might be attached to another protein, which in turn changes its conformation or changes its type, right? So, for example, proteins like ubiquitins like to bind to the protein and send it somewhere, right? So if we
I love that name, by the way, ubiquitin.
if we are able to develop these different probes to different forms of the protein and different conformations and different binding sequences, then we might increase the likelihood of finding what this bad guy is,
Yes.
and what the bad guy's signature is, and the multiple signatures of the bad guy.
Yeah.
That makes sense.
Totally.
Oh, good.
So you essentially have to. Are you screening for 100 different conformations?
We don't have
different shapes?
100. So
you
we
don't need the probes to go hunting.
So we
Huntingtin.
recently profiled 12 that seem to be top candidates used in animal models of disease, as well as in vitro in a petri dish model of a disease. So we profiled 12 and we found 3 good candidates. So those 3 good candidates, what we've now discovered, label different chunky conformations. I use chunk because it describes a big bulky protein, right? Different chunky conformations of the protein. And so what we're now trying to do is work out what binds to certain chunks of the protein. Perhaps we then can help work out this molecular signature of what the bad guy looks like.
Yeah.
So that's kind of what I'm trying to do now.
It's like you're a molecular neuroscientific detective trying to figure out who the bad guy is.
Kind of.
Needle in a haystack?
That
Does it feel like
sounds.
that?
It does. It does. I think we do get a lot of information from people doing work in cells in a dish, because if you throw Huntington into cells in a dish and it does different things, you can kind of work out molecular pathways to target, which is quite helpful for us. But yeah, it is a little bit like that. I think it's slightly better than a needle in a haystack.
Yep.
I think it's more like 100 needles in a haystack. We do have
Well, how
targeted
big's the
pathways.
stack?
That's right. It could be a very small stack, isn't it? But coming back to it, I think we're getting closer.
Yeah.
I have confidence in this. I feel like we're making good headway, and I think this is why, in all honesty, if we cure Huntington's, I'll be super stoked because I'm just going to lie on a beach and probably retire early because I'd be like, this is just incredible, right? But until then, we're going to keep going here until I can retire. And I would love early retirement. That would be incredible. Yeah.
But I think a lot of this will inform and educate people on really the true complexity on a lot of the things that are yet to be solved. I mean, proteins don't do one thing. No. There's not a linear pathway between gene, mRNA, protein, task that protein does. Like, it is an absolute web of... And then don't even get me started on like the other things in the dark, the DNA dark matter. Like there is a lot of things that don't code for protein that also do things. So, I mean, I find it absolutely fascinating. It's like we think that we're on the cusp of solving everything, but we don't realize how complex we truly are, and then also how nested within our own true origins and biology we are as well. Like we can have all the knowledge in the world, but we also need to apply the wisdom of the past as well.
That's right.
It's amazing. But in your other work that you do, so you work with, so it's Huntington's Disease Organisation, you started that?
Yes, that's the Huntington's Disease Youth Organisation of New Zealand. It's the New Zealand branch. Right. We operate under the international Big Sister organisation, but we support youth in New Zealand that are at risk of Huntington's, may or may not have the gene, but certainly come from a Huntington's family. And so our mission is to advocate, support, and educate those that are impacted by Huntington's, because you can be a young person, you may not have the gene, but you may end up becoming a carer for a parent. Or you might be a young person, you might be at risk, you may or may not want to get tested for the gene. That's a huge life-changing decision. So the organisation is widely focused on asymptomatic individuals that are sort of under the age of 35, because those who are symptomatic are under the care of the national and regional associations. So there's like a clinical body that looks after them. But my sort of area is more focused on the youth, because I feel like there was a big gap in the system in terms of supporting them, because Huntington's is a rare condition, there isn't a lot of government support that comes into supporting people with this condition, and it is rarer, right? So where do they get the support from? So this is how the youth organisation was actually born. It's quite an interesting story, actually. I was presenting at a national conference in 2013, and I was sharing some research findings from our post-mortem human tissue studies. And at the end of my talk, a member of the community, and this is what I love about our patient community, they really tell it like it is, and they can really get us to pull our socks up. She stood up and she said, yeah, all fair and well, you're studying brains from our families, and you've got all these findings. What are you doing for my kids? And for me, that was a real sobering moment. And I also felt quite humbled by the fact that someone from the community asked that of me, because obviously they look to us for answers. They look to us for direction, and suddenly I had no words to even help this particular woman who obviously has children. And so there were other members of the Huntington's community in this conference, and one of them, her name's Leanne Knox. She comes from a family with HD. She came up to me and she just said, hey, why don't we start a support group for kids? And I'm like, what? I don't know how to do. I don't have the skill set for that. I don't have expertise working with living people. I work with postmortem. It was I was quite taken aback by it. And she goes, I'll help you. And Leanne and I teamed up and just started a small support group. It started off as just a small group. We had a little website that we created, and we were just trying to get youth to engage with us. And we're trying to find out the need across New Zealand of where pockets of youth are that don't necessarily have support. And it kind of exploded, and we became a registered charitable trust. We now have representatives across New Zealand. We now have sort of really made links with the associations across the country, as well as the genetic counselling support services. And now we're on the verge of launching a counsellor program. Because one thing that the youth have said to us is, irrespective of whether we test for the condition or not, we don't seem to get a lot of counselling support. And that's just simply because, as we know, New Zealand as a whole has quite a pressurised healthcare system. And so having counsellors available to support youth that's funded, especially if they're considering testing and even after the result, there aren't many sessions available to that person. There's a minimum number of sessions. If you test gene negative for the disease, there really isn't a lot of follow-up, maybe 1 or 2 sessions, depending on the psychiatric status of that patient, that client. And so we've now set up a counselling support service, and that was probably, for me, one of the highlights of my career, actually, because as a researcher, we're always trying to make a direct difference. And this was my way of saying, okay, while I work on these therapeutic options in the lab, the cure may not come tomorrow, but at least I'm doing something today. And that for me is kind of, that's the reason why I wake up in the morning for these families, is because I know that there is a group of youth out there that are going to become prospective carers. They're going through a hell of a lot. And at least we're doing something directly to support them. So HDONZ is my cup-filling charity. And as a researcher, that sort of gives me confidence that we're taking the knowledge that we have and injecting it back into the communities that need it. So I demystify my science blog back to those communities, so we have our information delivered regularly to them as well. So that's why HDONZ is always going to be very close to my heart. And if, for any sort of public engagement I do, I don't ask for money, but I say, hey, if you want to support a good cause, let's support the youth impacted by Huntington's.
Amazing.
Yeah.
Well, I love it. I love your work. Really inspired by your absolute passion for what you're doing. It's quite amazing, and it's palpable how much energy and effort you put into the potential cures of tomorrow, but also the status of people today dealing with Huntington's, either directly or indirectly. Where can people, just to wrap it up, where can people go to find out more about you, what you're doing, what's next on the cards for you, if they want to get in touch?
So there's lots of ways to contact me. You'll find me through the University of Auckland. So if you just literally Google my name, you'll see my university profile come up, and my email address is there. A lot of people contact me through there. You can also find me on LinkedIn. If you're wanting to know more about the Huntington's Youth Organisation of New Zealand, just go to www.hdo.co.nz, hdyo.co.nz. But yeah, I'm always around. It's not hard to find me.
Excellent.